Hello everyone, myself Dr. Anuradha Sharma, and today we will do a crash course of most important points of biology for the upcoming NDA exam, which is to be held on 3rd September 2023. And this is a small introduction of mine, that is I am having 15 years of teaching experience and I am ex-faculty of STC quota. I have done my PhD from PGI Chandigarh and I have cleared GATE, UPGATE, CTEM and many other competitive exams. We will start this study with a small quote, that is, arise, awake and stop not till the goal is reached. This is my icon of the YouTube channel Dr. Anuradha Biology Classes. Now in this crash course we will discuss the all important points which are to be coming in your NDA exam. So we will discuss only the important points because we cannot cover the entire biology in this crash course. So we will just cover the important points. So first topic which we have to cover is the cells. As we know, cell is the most important topic. So we will cover the important points of the chapter cell. What do you mean by cell? We know cell is the fundamental unit of life. And we know the word first living cell was discovered by scientist Robert Hooke. This is also important. Okay, first living cell was discovered by scientist Robert Hooke. Now, cell theory is important. Cell theory is given by scientists Sweden and Shan. Two scientists gave the cell theory in the year 1938. Now, this cell theory gave two important points. First, all the living organisms, they are composed of cells and they are products of cells. And all these cells, they arise from pre-existing cells. All the cells, they come from some of the pre-existing cells. That means cells were already there and these cells give rise to another cells. There is another word for this, that is cellular, cellular. So this is important. Cell theory was given by scientists. Two scientists gave the cell theory. Now there are some important points. You should know smallest cell. Which is the smallest cell? Smallest cell is mycoplasma. It is also known as PPLO. That is pneumonia like organisms. So sometimes they can ask mycoplasma and sometimes they can ask PPL. You should know both the names. Smallest cell is PPL or mycoplasma. Now what is the largest single cell? Largest single cell is ostrich egg. You should also know longest cell. Longest cell is nerve cell. It is also known as neuron. Size of bacteria is 3 to 5 micrometer. Human RBC, it is 7 micrometer in diameter. Now fluid Mazak model. It is the model of double membrane structure. Cell membrane. So cell membrane, a particular model was given. It is widely accepted model and it is known as fluid mosaic model. Singer and Nicholson were the scientists who gave the fluid mosaic model, and it was given by in year 1972. Now, according to this model, quasi-fluid nature of lipid is there, and in this quasi-fluid nature of lipid, there are proteins which are floating. So this is again important fluid mosaic model. Now there are two types of transport. One is active transport and one is passive transport. You should know the difference between the active transport and passive transport. Now in active transport, the main difference between the active and passive transport is that in active transport, basically the transport involves expenditure of energy. In active transport, energy is required. Whenever there is transfer of any material across the membrane, energy is required, and energy is required in the form of ATP. So this transport is occurring across the cell membrane. So active transport, basically it is a transport whenever energy is required to transfer a material across the cell membrane. Whereas in passive transport, the cells do not require any energy. So in passive transport, there is no energy. In active transport, it occurs across the concentration gradient. And last, it is a rapid process. Active transport, it is a rapid process. Why it is a rapid process, whereas passive transport, it is a slow process. Now we have to clear this point that active transport is a rapid process. You have to understand this point. If we will, if energy is required, like ATP is required in active transport. So if we will apply any energy, any force in this active transport, if we will apply energy, any force, then definitely that work will be done faster. So in active process, we are applying energy. So definitely this active transport will be rapid. Whereas in passive transport, we are not applying any energy. So this is comparatively slow process. Now another thing we should know is gram staining. Gram staining, it is a technique to stain various cells. Now we know that there are certain cells, there are certain microorganisms which we cannot see by naked eye, because there are very minute microorganisms, smallest microorganisms. We cannot see these microorganisms by naked eye. So how we will see these microorganisms? We will see these microorganisms with the help of microscope. Now microscope, there are different types of microscope, like compound microscope, like light microscope is there. All the microscope has different resolutions depending upon the substance which we want to see. Now for microscope, we cannot just place the substance like that. We cannot just place the substance under microscope and we can directly see that substance. We have to stain that particular substance or cell before seeing under microscope. So a particular process is there, like gram staining. For this gram staining, it is only for the bacteria. If we want to see bacteria under microscope, so we have to stain these bacteria with gram staining. Now this gram staining, it was developed by the scientist Christian Gram. He developed this gram staining method. Now this gram staining, it was based on the envelope, that is thickness or cell membrane. So gram staining was based on the cell membrane. So bacteria, we know basically bacteria are of two types. One is gram positive and one is gram negative. So we know that gram positive, they are those bacteria that take up the dye, that is gram stain. the bacteria which take up the gram stain, they are considered as gram positive, and the bacteria that don't take up the gram stain, we considered as gram negative. So based on the cell envelope, this gram staining was developed. So we can say whether a particular bacteria is gram negative or gram positive. So this diagram you can see, if the bacteria is gram negative, then it will give, definitely it will give pink color. It will show pink color, and if it is gram positive, it will show blue color, like Staphylococcus, it is gram positive, and E. coli, it is gram negative. So it is showing this pink color. So this is gram staining method. You should know it is, what is the basic principle of gram staining method. This is important. It is based on the cell envelopes. Now in cell chapter, we should know different organelles are there. So we should know the function of each and every organelle. First we should know lysosomes. What are the functions of lysosomes? Lysosomes are basically, they contain hydrolytic enzymes. They are membrane bound vesicles and they contain hydrolytic enzymes like lipases, proteases. What is the function of these lysosomes? They digest the carbohydrates, proteins, lipids and nucleic acid. So inside lysosomes there are hydrolytic enzymes. They help in digestion of carbohydrates, proteins, lipids and nucleic acid. Now next important is the mitochondria. Mitochondria is the most important cell organelle. Why it is important? It is, the question comes from mitochondria in most of the competitive exam. Why? Because it is the most important cell organelle. Why it is most important cell organelle? Because it is known as powerhouse of cell. It is, why it is known as powerhouse of cell? Because it is providing energy. It is providing energy to the cell. And how it is providing energy to the cell? It is providing energy to the cell in the form of ATP, which is known as, ATP is known as currency of the cell. So, as we use generator in our houses to get energy, similar is the work of the mitochondria also. It is providing energy to the cell. These are the sites of aerobic respiration and they provide energy to the cell in the form of ATP. They are double membrane structure and enfoldings are there which are known as cristae. And these have also specific enzymes. So these are, mitochondria are very important. Now next is George Pillet. They observed ribosome. Ribosome in the year 1953. What is the function of ribosome? Ribosome, this is the structure you can see. These round structures are ribosomes. Now ribosomes, what is the function of ribosomes? Ribosomes are known as protein factories of the cells. Ribosomes are considered as protein factories of the cells. So what is the function of ribosomes? They help in formation of protein. Protein synthesis takes place. So these ribosomes, they help in protein synthesis. And ribosomes, basically there are two types of ribosomes. In prokaryotic, 70S ribosome is there, and in eukaryotic, 80S ribosome is there. 70S ribosome, it is further divided into two subunits 50S and 30S, and 80S ribosome is divided into 60S and 40S. So these are the two subunits of the ribosomes. So functions of ribosome, I have already told you that these helps in protein synthesis. Now next structure is nucleus. Now nucleus was discovered by the scientist Robert Brown in 1831. So nucleus is very important. It is known as brain of cell because it is controlling all functions of cell. So nucleus is known as brain of cell. You can see this is nucleus, this is the DNA material, this is chromatin, ribosome is present. So this is the scientist Robert Brown, he discovered nucleus in 1831. So the material of nucleus was stained by basic dyes and it was given the name chromatin by Fleming. And nucleus has some cells, mammalian RBC is there. Now on the basis of inside the nucleus genetic material is present. We can see that inside the nucleus what is present inside the nucleus? Inside the nucleus chromosomes are present, and chromosomes, these are the chromosomes, these are present inside the nucleus, and inside the chromosome genetic material is present. This is known as gene, and gene has basic material which is known as DNA. Okay, so now we need to know what are the different types of chromosomes. So chromosomes are basically divided into 4 types on the position of centromere. Now what is centromere? This is the centromere which is present in the center. These are arms. These are the arms of chromosome. A chromosome is having 4 arms. 1, 2, 3 and 4. So basically 4 types of chromosomes are there on the position of centromere. Now this is metacentric, means centromere is present in the center. All the arms are equal. When all the arms are equal, it is known as metacentric chromosome. Now some metacentric means 2 arms are bigger and 2 arms are shorter. So it is symmetric. Acrocentric means 2 are small and 2 are big. And telocentric means 2 are very, it is the centromere is present at the end. When the centromere is present at the tip, it is present at the end. When the centromere is present at the tip, that is end of the chromosome, it is known as telocentric. So basically 4 types of chromosome are there. So chromosomes have non-straining secondary contraction at the certain location, and this is known as satellite. So DNA, we know what is DNA? It is a genetic material, and it is responsible for the transmission of characters from one generation to another. All the hereditary characters are transferred from one generation to another through this DNA. And gene, what is gene? Gene is a segment of, it is a DNA segment. So it is a basically DNA segment which is responsible for transmission of characters. And for one single character, there is a one gene responsible in our body. So all the gene, if there is all the genes in the body, it is known as genotype. So genotype is the genetic makeup of organism, and phenotype is the external characters. External characters because of genes. So the external appearance of organism because of these genes is known as phenotype. So this you should know. What are the different 4 types of chromosome, what is gene, what is DNA, what is the function of DNA, what is the function of gene, what is genotype, what is phenotype. And one more term is there, what is it is allele. Allele is alternative form of gene. Sometimes it is asked, define allele, or what is allele? Allele is alternative form of gene. So these are the terms you should know. Now we should know some differences between the plant cell and the animal cell. Basically in plant cell, this is the diagram of the plant cell. You can see this is plant cell and this is animal cell. Now in plant cell you can see cell wall is present. Where is animal cell? There is no cell wall. In plant cell plastids are there. Plastids are present in plant cell. Whereas in animal cell no plastids are there. In plant cell large central vacuole is there. Whereas in animal cell there is small vacuole is there. In plant cell centrioles are absent, whereas in animal cell it is present. So these are some of the differences between the plant cell and animal cell. Now you should also know what are the differences between prokaryotic and eukaryotic cell. Prokaryotic means it is the initial cells, old cells, and eukaryotic cells means new cells. So in prokaryotic cell these are generally smaller, and eukaryotic cell it is larger. Now in prokaryotic cell there is no nucleus. As there is no nucleus, no nuclear membrane is there. Whereas in eukaryotic cell, nucleus is there and nuclear membranes are also there. Now in prokaryotic cell, circular DNA is there. In eukaryotic cell, linear DNA is there. In prokaryotic cell, ribosomes are 70S. In eukaryotic cell, it is 80S. So these are some of the differences between the prokaryotic and eukaryotic cell. Now this is the table showing the scientist, year and the work done. This is also important. You should know in which year the scientist has done which work. Which scientist has done which work. Now in 1665, Robert Hooke, he examined the cork sac under microscope. In 1650 and 1700, Leeuwenhoek used simple lens and observed bacteria. In 1831, Robert Brown discovered the nucleus. In 1838, Schiedel, Shaw and Rudolf Virchow, they proposed the cell theory. This is important. In 1840, Purkinje gave the term protoplasm. In 1898, Camillo Golgi discovered the Golgi apparatus. In 1953, Palade observed ribosome, and in 1972 fluid mosaic model of cell membrane was discovered by Singer and Nicholson. Now this is the table showing the scientists and the name which has been given to them. Like Aristotle is known as father of biology. Theophrastus was known as father of botany. Aristotle again known as zoology. Father of plant physiology is Stephen Hales. Father of taxonomy is Carl Linnaeus. Father of genetics we know is Regan Mendel. Father of experimental genetics is Morgan. Father of biology is John Hedwig. Father of modern cytology is Sorensen. Father of microscopy is Malpighin. Comparative anatomy is Cuvier. Father of experimental physiology is Gallian. Now father of eugenics is Galton. Father of Ayurveda is Charaka. Father of bacteriology Robert Koch. Hugo de Veres is father of mutation we know. Like father of immunology, Edward Jenner is known as father of immunology. Lance Steiner is known as father of blood groups, father of taxonomy, Santa Pau. So this is also very important. So just please subscribe my channel and press the bell icon so you can get more videos. This is part 1 of this, and soon I will be bringing part 2. So you can just practice for your exam, and I will be bringing part 2 of this video.